Welcome to day 2 of explaining something to understand it better, also known as the Feynman technique. Now I will talk about the steps during NGS. I've already spoken about what we do before NGS to get DNA or RNA in another video. This is to explain the steps during NGS. So the first step here is fragmentation. Whatever DNA or RNA that you have is very long, and every instrument that you use, NGS instrument, has a particular capacity to sequence. So you break down DNA or RNA fragments according to the instrument you're using. Second step is end repair. Since you're breaking the DNA, the ends are obviously broken in a weird, abrupt way, so you have to repair those ends. Third is ligation of adapters. So you attach something to the ends that helps you mark the DNA fragment. Fourth is size selection. Many fragments, different sizes, you select a particular size according to a particular instrument. Fifth is immobilization. All of these sizes, all of these strands, you kind of stick them at a particular place. That is immobilization. Sixth is amplification. Since you want to be sure of the base that you're reading, alright, whatever signal that you're getting to an instrument, so the same DNA fragment, you basically make many copies of it, so that whatever result that you get, you can be absolutely sure that this base is present at this position. And the seventh step is obviously sequencing, to understand the sequence of bases in DNA or RNA. Now I'll talk about each of these steps in detail. So the first step that is done during NGS is fragmentation. Like I said, this is done because every instrument has a particular length of DNA or RNA that it can sequence. So first, an enzyme called shearase was used to break DNA. Shearase is an endonuclease that randomly but uniformly cuts bonds in double-stranded DNA. This is controlled by concentration. So more enzyme, more cuts, smaller fragments, less enzyme, less cuts, larger fragments. To remember this, a simple analogy is imagine you are cutting a carrot. The more cuts that you make, the smaller pieces that you get. And if you make less number of cuts, you'll get bigger pieces of the carrot. Shearase works in a similar way, but for DNA. Restriction enzymes are actually not preferred for fragmentation because restriction enzymes depend on a particular sequence that they can recognize and cut. And because of that, the length is varied. It depends on the number of recognition sites. So it cannot be controlled as well as shearase. And since we can control the number of cuts by changing the concentration of shearase, this difference in length is very important since it can be varied, and it also ensures that all parts of the DNA are represented. But even shearase does have some disadvantages. For example, shearase does not have reproducibility. In different experiments, if different concentration of shearase is used, then we'll get different fragments of DNA. So another method called sonication is highly preferred and highly used. This is the mechanical DNA fragmentation based on cavitation. In the previous video, I have spoken about this. So it is similar. High frequency sound waves are used, and this is reproducible because all the parameters in a sonication experiment can be used again in a similar way in another experiment. The second step is end repair. So after fragmentation, we may not get homogeneous blunt-ended fragments, which means that both the DNA strands may not be cut at the same base. DNA strand 1 may be cut at base 4, DNA strand 2 may be cut at base 6, which will give us overhanging ends, sticky ends, which we don't want for sequencing. That is why we do end repair. The third step is ligation of adapters. DNA ligase is something that joins two DNA fragments, and we use DNA ligase to attach an adapter to the end repaired DNA sequence. Why we attach adapters? There are a few reasons. So one, adapters are known sequences. So they help us identify. They act like a barcode or an index. So for example, if you are sequencing the DNA of 3 or 4 organisms together and you want to be able to differentiate which organism's sequence is the result that you're getting. So to do that, you attach adapter number 1 to organism number one's DNA, how many ever fragments that you have. You attach adapter number one to all the sequences of organism number one. Similarly, adapter number two to all the sequences of organism number two, and so on. So that helps it act like an index, a barcode basically. And also, adapters act like complementary sites to oligonucleotides on a surface that we will use in the next further steps for immobilization of DNA. Simply put, these DNA fragments have to be stuck to some slide, to something. Depending on different machines, there are different things that we stick the DNA to. To stick these, of course, we need a complementary DNA fragment so that we can stick this DNA fragment. So adapters help by acting like this complementary fragment. The fourth step is size selection. So to select DNA fragments of a particular size, one thing that can be used is gel electrophoresis, but it results in the loss of a sample, so it is not used. I'll still explain in one simple line how gel electrophoresis is used to select a DNA fragment of a particular size. So sample is taken, DNA sample, and it is run through a gel. With it, a ladder, that means a sequence of known length, is also taken and that is also run on the gel. And based on migration of the ladder and the DNA fragment, a DNA fragment of desired range can be selected. And this also helps eliminate unligated adapters. So like I mentioned in the previous step, step number 3, ligation of adapters, extra adapters are added so that two adapters can attach to a sequence to increase the probability. But because of that, there will be some adapters that are left in the solution, or two adapters that have binded together, or maybe two sequences that have binded together. So to remove all of these extra sequences that we do not want, this would work. But since it results in the loss of a sample, there is another technique that is used known as SBRI based size selection, which stands for solid phase reversible immobilization. So here beads are used. These beads are made of polystyrene with a layer of magnetite and carboxylate. The beads are paramagnetic, which means that they are magnetic only in the presence of a strong magnetic field. DNA binds to the carboxylate on the bead only in the presence of PEG and NaCl. And here the ratio of number of beads to the volume of the sample determines the size range, which means if you use less number of beads, only long DNA fragments get attached because they have more charge and get attracted strongly by magnetic field. So DNA has some charge. The longer the DNA, more the charge. So that's why longer DNA gets attracted more easily to less number of beads. And if there are more beads, then even the smaller DNA fragments get a chance to attach. The fifth, sixth, and seventh steps are immobilization, amplification, and sequencing. I have not explained these three steps in the day 2 video because these three steps are different for each different NGS machine or instrument that you're using. So in the next video, when I'm explaining NGS instruments and machines, I will explain these three separately for each of those machines or instruments.